Hello, good evening, everyone. Today we will see what are all the things, especially now. Today we will see, especially the chapter 4 or the unit 4, animal genetics and breeding, heterosis and mutation in animal breeding. And this is the overall class, 39th class is running, and in the animal genetics and breeding, it is 5th class, and it is dealing with the heterosis and mutation in animal genetics. So, this heterosis and in animal breeding is especially for understanding the hybrid vigor of an animal. Hybrid vigor, or what are all the uh, advantages of hybridization of the animals. So this will be dealt in this particular chapter is concerned. Hence, we will see the things one by one very fastly. Then, as the introduction, heterosis is also known as hybrid vigor, which is a phenomenon in which the cross-bred offspring show superior traits when compared to the pure bred parents. So this concept was first coined by George Shull in 1914 and has since become a fundamental aspect of the animal breeding. And heterosis plays a very crucial role in enhancing the traits such as growth, reproduction, and overall fitness of the particular animal, and making it highly valuable for agriculture and livestock industries. So the importance of the heterosis is like this. The significance of heterosis lies in its the ability to improve both economically important traits, and also the ability of the offspring to produce the required things to the offspring. So overall animal productivity will be increased and also the reproductivity. And by over aging the genetic diversity, breeders can produce offspring that exhibit increased vigor, better reproductive performance, and vigor adaptability to environment conditions, different environment conditions. So this is the heterosis is very much important in developing the crossbred animals. Then it offers an array of advantages, especially it has got the advantage of lovely heritable traits. Then heritability is the proportion of variation observed in animals due to the genetics passed along with the generations and the phenotypic variation observed due to the genetic function of environment, that's the effects. So the types of the heterosis are, varieties of the heterosis are there. The number one is the individual heterosis. So it is the improved performance of the genetic trait and benefits of the crossbred animals. So it is the improved version of the genetic traits. Then here the second one is the maternal uh, heterosis, means it is the benefit observed in crossbred mothers such as higher calving rates and improved weaning weights. So this is a little bit change in case of the individual heterosis, wherein with the improved performance of the individual crossbred animals is seen as one of the criteria. Then the maternal heterosis is the benefits observed in the crossbred mothers. It is not the offspring, such as the higher calving rates and improved weaning weights, etc. Whereas the third one is the one where in which the maternal heterosis is enhancement of enhancement of the reproductive traits in crossbred sires. So this is the third type of the heterosis is concerned. Whereas all these three differ a little difference from one another. So the genetic theories explaining the heterosis are different. The number one is the dominance theory, and it is the superiority of hybrids due to the dominant alleles, as I have yesterday explaining, suppressing undesirable or recessive alleles. Whereas the overdominance theory is another theory wherein which the heterozygous individual exhibit superior traits. So this is the second theory, whereas the third theory is the epistasis theory. The third theory is the epistasis theory. So this is also another theory wherein which the epistasis means interaction between genes at different loci. It results in enhanced effects of the particular trait is concerned. And the benefits of the heterosis in case of the livestock production. What is the benefit? You might have heard, especially in case of the undergraduate, but always we forget it. So better to learn it once again. Here, the enhanced growth rates lie the increased weaning weights and overall body size will be there. And for example, here in the right side on photograph is there a bull of a breed A which performs 100%, and the cow of B breed. It also performs 100%. Then the F1 crossbred progeny will have the performance of A 50%, then performance of the B with 50%. So always it will be a improved reproductive efficacy will be there, and the traits such as fertility and longevity are always enhanced. Whereas the greater resistance to the disease is another thing. Whereas the crossbred animals show stronger immune response as they do contain the blood of the local animal. Then there are a lot of studies on heterosis have gone during the last 100 years, and crossbreeding systems can increase calf weaning weights by up to 20% and extend the productivity of the span of cows by over a year. Hence, the crossbred dairy cattle also produce more milk when compared to the purebred counterparts, when compared to the feed intake, and also the sustainability or resistance to the different diseases like mastitis, foot and mouth, and many more diseases. Are also the diseases like the ketosis and other things, metabolic disorders. Then there are a lot of challenges are also there in this. The benefits of heterosis are most pronounced in the first generation itself only, F1 hybrids, whereas subsequent back crossing can dilute these effects, requiring careful management and also the planning. So the heterosis is referring to the superiority of the F1 hybrids in one or more characters over its parents. So the term hybrid vigor is used as synonym for the heterosis is concerned. So it's a synonym, especially the heterosis is concerned, and we can say it take or it's for the, but it differs from the luxuriance, and heterosis has increased F1 over parents in general vigor, yield adaptation, then luxuriance has increase of F1 over the parents in vegetative growth, but not yield the adaptation, especially if it comes to the crops. Then the hybrid vigor, according to the shell, the developed superior to the hybrids is the hybrid vigor, and heterosis is the mechanism by which the superiority is developed as per the crossbreeding program is concerned. So here there are historical aspects of the heterosis are written in 1766. This started as first pre-Mendelian things are there, and it happened in such a way that the heterosis made a very strong basis for the crossbreeding. And 1799, 39 principles of anti-inbreeding is also there. And 1865, Mendel, he informed the hybrid traits and also the vigors. Whereas in case of 1877, Charles Darwin, he showed cross fertilization is always beneficial. In 1880, Beale is the first published report of 51% increase in the yield of the offsprings when compared to the genetic parents. In 1891, Johnson informed the crossing gave better offsprings, whereas in case of 1892, McClear inbreeding imparted sterility, crossing impaired vigor is also another thing. Whereas the history of heterosis concept goes on as Shell once again in 1914 coined the term heterosis, and 1918 Jones he improved the practical utilization of heterosis. Jones 1918 doubled the cross. Then Franz Lamar in the 1800s, Correns divorce in 1900 and other things. So it went on, and recently also so many concepts of the heterosis have been introduced both in the plant and also in case of the animal kingdom. Then, Lord, this hetero Beltiosis is coined by Blitzer in 1968. So it is a term refer to the increased performance of hybrid over the BP or the original line. And euheterosis is the hybrids possess higher fitness than that of the parents, whereas the luxuriance is an extreme heterosis for morpho characters but no fitness is another thing, whereas the positive and negative heterosis is referred to include majority of the hybrid over parents adaptive heterosis. So it is also called as the heterosis for adaptability. Then types of the heterosis have got two types. It's also called as euheterosis or true heterosis, means superiority of the F1 hybrids over the parents. Whereas the pseudo-heterosis is the luxuriance, is an overexpression or quicker growth of the characters in a variety of the species, especially hybrids, which may not result from either masking of deleterious genes or from balanced gene combinations throughout the hybrid. Therefore, the luxuriance is overexpression of a trait or temporary excessive vegetative overgrowth of a plant, or usually it happens in the plant which has no adaptive significance. So it's a temporary thing. Whereas on the basis of types of estimation of the heterosis, the average relative heterosis, then heterobeltiosis, useful or standard or economic heterosis are the three classifications are there. So there are varieties of the formula are there to calculate the average heterosis, which is the heterosis where F1 is superior to the mid parent value. In other words, superior to average of two parents. Then average heterosis where F1 means the hybrid, MP is equal to mid parental value, P1 plus P2 where P1 is equal to parent 1 and P2 is equal to parent 2. And this type of heterosis is of no use in agriculture since the superiority is below the better parent value, and it is just calculation. Similarly, the heterobeltiosis, where with the heterosis is over the superior parent of superiority of F1 over the better parent, either 1. And economic heterosis or useful heterosis is calculated by the superiority of the F1 compared to the high yielding commercial check variety in a particular crop or in case of animal. Whereas the negative heterosis is totally opposite. It is the performance of F1 inferior to the better parent or mid parent value with the duration. And genetics basis of the heterosis by is still following the three hypotheses. Number one, the dominance hypothesis. The B is the over dominance hypothesis, whereas the another one is the epistasis hypothesis. Whereas the dominant hypothesis always say it is a heterosis not due to the heterogeneity but dominance hypothesis. So this is the superiority of the dominant alleles over the recessive alleles in a simple term, but the heterosis is due to masking of deleterious recessive alleles by dominant alleles. And heterosis is directly proportional to the number of dominant genes which is contributed by each parent. So, suppose the parent 1 is contributing to the offspring, then the parent 2, then it is called as the dominance hypothesis of the heterosis. For example, the parent has got the alleles double A, small double b, capital double C and DD, whereas the another parent has got the opposite thing. Then the phenotypic value will be 2 minus 1 minus 2 minus 1 is equal to 6. So this is the F1 generation. So there is a lot of debates went on the dominance hypothesis. So many times the Henry Jones he also informed that F2 curve should be skewed towards the dominant gene and dominance of the linked gene hypothesis in the Jones 1917 he proposed. So there may be a linkage between some favorable dominant genes and some unfavorable recessive genes, and as a result, it is not possible to obtain true breeding homozygous individuals for all dominant genes. So these are all some of the explanation given, and over-dominant hypothesis heterosis is also proposed by Shell and East in 1908, supported by many other scientists also. This theory is called by various names. For example, stimulation of heterozygosis, number one, cumulative action of divergent genes, alleles, single gene heterosis, superdominance, and overdominance. So, according to this hypothesis, heterosis is a result of superiority of the heterozygote over its both homozygous parents. So, superiority of the heterozygous may be also arise due to the production of superior hybrid substance in heterozygote than cumulative action of divergent alleles. So the third one is the epistasis hypothesis. It refers to interaction between alleles of two or more different loci. It's also known as non-allelic interaction, and non-allelic interaction of three types like the additive X dominance, dominance X dominance. So the epistasis hypothesis is mostly attributable to favorable epistatic interaction between non-allelic genes. So, out of above three genetic hypotheses, the heterosis is the dominance hypothesis of heterosis. The dominance hypothesis is most widely accepted. Please remember that about the three things, the important thing to remember is hypothesis of the dominant hypothesis. Heterosis of the dominant hypothesis is the most widely accepted hypothesis. So overdominance and epistasis also operate in the manifestation of the heterosis. Whereas the fixation of heterosis is also done by asexual reproduction, then apomixis, then balanced lethal system, and also by polyploidy. For example, in different crops, this experiment has been done. Then the manifestation of the heterosis is maybe in the following form: increased yield, increased reproductive ability, increase in the size and vigor, and better quality and greater adaptability is also there. So other than this, then there are varieties of the other facts like mode of pollination, then genetic diversity in patients, then genetic basis of the patients, then adaptability of the parents is always referred to the plants is concerned, whereas it is not so applicable in case of the animal genetics is concerned. Just to know certain terminologies, we have to look into this matter. So, the development of hybrid varieties. Suppose we want to develop varieties of the hybrid like Giriraja, Girirani, then the crossbreds. So we need to understand all these things. Then the practical of this hetetosis is fully exploited in the form of the hybrid animals, especially in case of the plant genetics, partially in the form of synthetic composite varieties. So the important factors in commercial exploitation of the heterosis is enough magnitude of heterosis, high percentage of outcrossing, then floral biology, then availability of NS and SI. So instead of this, all these things, the crossing the inbreeds indigenous versus exotic origin result in the release of the best hybrids in plants breeding, not the animal breeding. Totally, the plant breeding is different than that of the animal breeding. Then, lot of varieties have been drawn like single cross hybrid, three-way cross hybrids, double cross hybrids, double top cross hybrids. Like this, they arrive. And single crosses, it's a simple thing, A into B, F1, SCH. Double cross means A into B into C into D. So varieties of the grasses and the thing, they are. So the most striking advantage of the heterosis is development of a crossbred which is yielding more uh, maybe milk yield or the crop yield than that of the parents. Then the modified single crosses, three-way crosses, double modified single crosses, and modified three-way hybrids. All these are later developed as one of the way. Then intervertebral hybrids have been also developed, top crosses hybrids, then interfamily hybrids, double top cross hybrids have been developed in case of the crosses. So the term mutation, then we are going to another term, especially in case of the genetics, is the mutation. So what is a mutation? It's a change in the genes, abrupt change in the genes, etc. So it breeding mutation, that's the Shudjan, was first coined by the Fredwen and Lane in 1944. So they refer the mutation breeding as deliberate introduction and development of the mutant for the crop production, and it is a milestone in the breeding. So 300 BC, the ancient Chinese book Lulan provides the first documentation of mutation section in plant breeding and other trace cereals in case of the China. And 1590, the first variable, verifiable plant mutant decide like incisa mutant of greater Britain. So chronologically, so many events happened, very similar to that of the mutation or inheritance, like in 67, then the 1907, extensive examples of the spontaneous mutants in crop plants have been uh, evaluated. So especially in case of plants, lot of research went on, but in case of the animals, it was not so fast. But because the lifespan of all these cereals or the wheat, and oats, etc., is very short. So the experiments could be conducted very fast, and new breeds were evaluated. So it went on. Since 2000 also, the development of tilling population were seen, which is one of the terminology that is used very properly. Then there are once again the terminologies like micro and macro mutations. So mutations are called as micro and macro mutations depending upon the magnitude of phenotypic changes or effect produced by them. So mutation is the magnitude of the change in the phenotypic characters. Then macro mutation produces a large phenotypic effect. Macro mutation. Then it is easily recognizable in individual plant basis. Then oligogenic in nature. Then can easily be selected as M2 generation. Then micromutation produce the small phenotypic effect and cannot be recognizable on individual plant basis. Then detected only in group of plants and need treatment of statistical data. Then apart from this one, the polygenic nature and selection delays till M3 are there. Whereas there are a lot of forces that changes the gene frequency here, then the forces here are the we can classify them as the mutation, migration, selection, and genetic drift. So this is also another important terminology. Already we learned the mutation, then migration, and selection. Whereas the mutation is the change in the base of the sequence of the genetic material, and some mutations occur at regular frequency, and mutation rate is low, and regular changes due to the mutation is very small. By chance, the some mutations end up making a difference in livestock, like the dwarfism in beef cattle in 1950. They invented that thing. Then migration means importing the new genes in a population or purchasing a new sire, opening the upbreed to new animals, then importing European breeds of the cattle, and it's a very powerful force in the gene frequency. Whereas the selection is the one where we choose the best young animal to be the best parent, then eliminating the inferior parents from the population. Progress is gradual but it is steady and should select on a balance of character. And genetic drift. Please note down this particular thing. What's a genetic drift? Genetic drift. So this is defined as change in gene frequency due to a chance. This is a chance. It's change in gene frequency. And each sperm and egg contain random sample of the genes from the parents. And sample may be above or below of the parents, and some offsprings are worse than average parents. So this is also another thing. Whereas the phenotypic variation is phenotype is equal to genotype plus environment. So usually the genetic makeup also very important. The it contributes about 75 to 85% of the thing. Then whereas the environment plays little less, and depending upon the food, etc., then it also changes. Then variance in the phenotypes, due to the variance in genotypes and environments, the especially the due to the variance in genotypes, the environment effects also play a very important role in the genetic drift. Then usually the difference between the genotypes may not be consistent, and all the environment, and difference between the genotype may not be consistent. For example, the Brahman crosses superior to British crosses in southern states, whereas the British crosses the superior to Brahman in other states, so northern states. So heritability is the one where in which the Heritability is proportion of phenotypic variation that is due to the genetic variation. So the this describes how easy to make progress through the properly if you select the sire and dam and maybe the value ranging from 0 to 1. Usually it is between 0 and 0.60. Then selection of the superior breed stocks is this type of technology is very much required. Then if available, individual performance is single most important piece of information. Then selection of individual performance is most effective in case of the heritability with high concentration. Then the selection of superior breeding stock is always important, like sibs, progeny, pedigree, and other relatives. Whereas some, especially for traits, low inheritability, and some traits not measured for potent carcass traits or traits measured only by one sex. Then predicting selection response. So what happens if I select a two good breeds? How to predict it is one generation of the selection is equal to response is equal to heritability into selection differential. Then selection differential is equal to difference between the selected to be parents and average group size. So usually this is also important for predicting selection response for several years it takes and also generation to generation intervals. Usually it takes long term. For example, swines it takes 2 to 3 years. Cattle it takes 4 to 6 years. It's not a easy job. So it can be calculated as heritability to selection differential generation of the interval. Genetic correction is the one where the selection of one trait cause genetic change in the another trait. That's called as the genetic correlation, and this is caused by the pleiotropy or the gene that affect more than one trait is called as pleiotropy. Then there are a lot of national performance programs are there. This was the need of uniform performance, especially in case of dairy programs. So they organized first beef programs followed, swine and sheep programs came later into this national performance programs in different countries. Also, dairy cattle performance programs. So the dairy herd improvement associations like Indian Dairy Associations and cooperative with United States Department of Agriculture or USDA. Then standardization location meant for using milk production for the 305 days. It is strictly followed there, whereas we need to adopt this one over at least in the long term basis. Then huge the genetic increase in the milk production over last 30 years has happened in almost all the countries, and hence the India stands number one in milk production. Whereas the beef cattle research is going very fastly in the other countries, whereas the guidelines for uniform beef improvement programs it takes place and establishes standard recommendations for measuring the growth efficiency, then also the reproduction. Then apart from this one, the swine also there were performance programs were there. National Swine Improvement Federation in US and India also lot of things were done to improve the swine production, especially in the northeast states. Then the US established standard recommendations for measuring the growth and recommends the indexes to use the selection. And same type of the genetic traits are followed in different parts of the country also or the world. And sheep performance programs, national sheep development programs. For example, in Karnataka State, sheep and wool development corporations are there, and they are slower to develop than the other classes of the livestock. Programs need to be very well organized, especially they are established for recommendations for measuring growth, efficacy, reproduction, then the carcass traits of meat producing animals. Then the every country will have the national genetic evaluation program because there is a problem how to make fair comparison with the parent and breeding stock raised in different environments because our country is a very diverse country from Kashmir to Kanyakumari, then also West Bengal to Gujarat, Madhya Pradesh, and so many states. There is a lot of environmental differences, water differences, then the environment where the cattle are being reared or cattle, sheep, goat, pigs they are reared also changes from the different states, one from the other. So what's the solution then? Use ties between herds that are established because many sites are used across several herds due to the artificial insemination. And I need to mention here that the artificial insemination program need to be strictly controlled and even though it is lot of genetic rules are there, nobody is going to follow, and hence we are getting lot of problems. Then national genetic evaluation program should be there, where with the breeder association they should maintain large database of performance record for their herd improvement program and data used to compare genetic merit of the animals to assess the different the breeds across the breeds and what are all the changes. Then national genetic evaluation policy is there for most of the countries, expected progeny differences, then the formula are there. Suppose there are bull A and B are there, whereas the weaning weight, especially expected progeny difference is plus 40 and B is 10. Then the means that a bull A is expected to sire cows that weigh about 30 pounds more than that of the cows from the side B. So these type of the calculations can be predicted like expected progeny difference. Then we need to evaluate the national genetic evaluation program itself using the expected progeny difference. It's called as the PTA of the dairy cattle or predicted transmitting ability. Then dairy conducted by the this is USDA. Then beef conducted by the Breeders Association. So there in different countries that doesn't mean that the government should interfere. Whereas in India, all the surveys and the breeding programs are totally controlled by the government of India. So the mating system is also classified as breeding, line crossing, then crossbreeding. Crossbreeding is one of the very important program going on. So inbreeding, what's inbreeding? Inbreeding depression. This is recessive alleles tend to be inferior genetically also. Then these cause decline in performance due to increase in frequency of recessive homozygotes, whereas most decline in reproduction in the development. Then mating system is line breeding. Then mating that causes large relationship to one outstanding ancestor while keeping inbreeding cow. So this is useful to retain the genes of outstanding individual who is no longer available for breeding purpose. And nowadays this is the frozen semen era, and even though the bull is not there, we can use these sperm straws or semen straws for artificially inseminating a cow or a buffalo. Then outstanding individual must appear in the pedigree or several times, at least 3 to 4 generations back. Then mating system is further it is continued as line crossing. It is the mating unrelated individuals within a breed. Unrelated means one ram is from XYZ farm, which is far away, and introducing the new ram. Then this caused some increase in the performance less than what is seen with crossbreeding program. Then mating systems continued. The crossbreeding, yes, it is a very one of the popular thing, and everyone knows this thing. and mating of the individuals from the different breeds of the cattle. Then here more and more heterosis will be there. Advantage of crossbreed individual compared to the awake of component of the breed. The F1 always performs better than that of the original breeds. So breed complementary is breed complementarity using profiles from the breeds while hiding the flaws. Then heterosis as usual, individual heterosis, advantage of the cross offspring. Then maternal heterosis, advantage of cross-bred mothers. Then parental heterosis, there is an advantage of the cross-bred, very good quality site. So all these things depend upon later ultimately end up with opposite inbreeding depression results from decrease in the heterozygosity, reproduction, large advantage of heterosis, whereas the growth, the heterosis plays moderate advantage from the growth and carcass little advantage. Then crossbreeding systems have been evaluated, and in every breed there is a crossbreeding. Then the terminal is specific breeds of sire mated to a specific breeds. So that's called as the terminal crossbreeding. Whereas the rotational crossbreeding is the breeds used in a regular cycle, daughters of one breed of sire mated to the next breed of the sire. So this also continues in breeding system. Then crossbreeding terminal uses maximum breed complementary, uses maximum heterosis, must bring replacement breeding stock every time. Then it should be a rotational process, replace the females retained by the system. And no breed complementary, and sometimes the loss in the heterosis may also happen. So, there are few of the conclusions we will draw it later, especially the heterosis is a valuable tool for enhancing the growth, reproduction, and overall performance of the livestock. So that's the advantage of heterosis. Then also there are a few of the disadvantages are also there, and we need to have the strategic crossbreeding program. Even though it's present in the country, it is not being implemented in such a effective manner as the against the breeding policy of the government. Lot of the other things which are unintended or intended we are being done, and because of this thing, the maintaining the pure breeds is one of the greatest difficulties. So we need to have strategic crossbreeding program. Then this has led to significant improvement in the productivity, especially in case of milk productivity and meat productivity in sheep and goat or other broiler productivity. So these are very well established things, especially using the pterosis. Then, yes, we are in the end of this particular chapter is concerned. Then, of course, this is time to thank you all, and have a great day. Answer the question papers every day. They are sent in a specific time, and they are always